glucose, a very simple blue plan figure, okay? Then going through TCA cycle. Okay. We skip all these chemicals. Hydrolyze 2 ATPs here, using 2 ATPs here. Generate 3 ATP for one branch right here, somewhere here. Multiply by 2, that is 6 ATPs. And just somewhere bottom here, generate 2 ATP for each branch. So, we have 2, that's 4 ATPs. Okay, so, in aerobic respiration, this total here is minus 2 at 10, 8 ATPs. Okay, pyruvate, go to acetyl-CoA, you have to know. Transition step, 3 ATPs. Okay, let's say I don't know any of these citric acid cycle. I don't care about all the chemicals. So what you should know? At least you know somewhere here there is an NAD reduced to NADH2. Okay, somewhere here there is another NAD reduced to NADH2. Somewhere here there is a FAD reduced to FADH2, and somewhere the cycle finished, there is an NAD reduced to NADH2. And don't forget, right in the middle of the circle bottom, we have the GDP generated become GTP. Okay, how many ATP are there? 3 ATPs, 3 ATPs, this is 1 ATPs, 2 ATPs, and that is 3 ATPs. So we add up them together. 1, 2, 3, 4. 3 ATPs multiplied by 4, add 2 ATPs, add 1 ATP. This is equals 15 ATPs. And then we multiply by 2 equals 30 ATPs. And then this guy and this guy add up together, becomes 38 ATPs. Is that good? That means if you don't need to do any extra points, that's the only thing you need to know in this class. Because what we care about is how many energy generated through the ATP process. Okay? Now, something else I want to mention. Why NADH reduced to NADH2 generates 3 ATPs? Why FAD reduced to FADH2 only 2 ATPs? This is the reason is the distance between the electron donor to the electron acceptor. NAD reduced to NADH2, the voltage value is more negative compared to the bottom one. So remember, we have NAD, go FAD, go FMN, FES, go COQ, go to oxygen. NAD, electron transfer, the distance is this large. FAD is only this large. Therefore, NADH generates more ATP than FADH. That's the reason. Okay. Now the second question is, going through aerobic respiration, theoretically there is a 38 ATPs generated. You know the bacteria can survive. And we talked about going through fermentation. Only 2 ATP generated. Then how come bacteria can still survive? Even with very limited amount of them. The reason is you need to go back to see one of the equations. is right here. This here is NADH2, oxidized to become NAD+. Once you see NAD+ there, once you see this guy there, which means there is some of the open space for this to come in. And this equals proton. What this means? Which means free available electron carrier is recycled. That is the reason why fermentation can be used by bacteria in their life cycle. Although it's only 2 ATP generated, because electron carrier is recycled. That's the key. That is the reason if one of the bacteria, imagine, only do glycolysis, they will not survive. Because you look at the glycolysis, the whole structure, the whole procedure, there is no free available electron carrier generated. That's why glycolysis cannot stop here. They either should go fermentation or they go TCA cycle. Which means either generate enough ATP, let them survive, or you generate limited ATPs, but your electron carrier is recycled. Which means there is a free available, this guy comes out. Okay, so that's something I want to mention. It's very magic stuff here for you to understand. Okay, so let's finish about here. And I said on next week, we will, on Thursday, we will go over the whole procedure again in the lab section to let you guys understand. Because that's also coupled with our lab work. We're doing the biochemistry test. This slide tells you how you calculate it, but be careful. Here is using 2.5 and 1.5. Okay, that's a new calculation method. That's why total is 32. In the exam, please using only one rule you feel comfortable. If you like the older one, this is the older procedure. This is what I'm writing here. I like to use the older one because 3.2 is easier to calculate than 2.5 and 1.5. And this is the older rule, so you can make a marker. This is the older rule, this is a new rule right here. Okay? Okay, next we very briefly talk about anaerobic respiration. Anaerobic respiration, we already talked about the procedure, which means electron acceptor is inorganic chemical, but is not oxygen. So what are the examples of the inorganic chemical? So many right here. Could be sulfide, carbon dioxide, ferric, Sicilian, and this right side here is reduced products. And right here is also the examples of the microorganism. So you see the disulfur vibro. When you see the name disulfur vibro, which means they use sulfide as an energy resource. And methyl-Gs, they use carbon dioxide as an electron acceptor. Final reduced product is methyl. And you go to the Pseudomonas, Bacillus, Geobacillus, they could use ferric as an electron acceptor. Okay? On and on. There are some examples here. But you need to know. Going through anaerobic respiration, the total ATPs generated is less than aerobic respiration. Because they are going through modified electron transport chain or some kind of modified citric acid cycle. The distance between the electron donor and the electron acceptor is much less than aerobic respiration. So it's not that much. This comes out with this a very good table. This table, I hope you study very well, will be a very good resource to review the material. Remember this user older rule, because it's a 38 ATPs. So aerobic respiration, glycolysis TCA cycle, electron terminal acceptance is oxygen, how many total ATP generates? 38. How many going through oxidative phosphorylation? 34. This is anaerobic respiration. generates 36 or less ATPs. Fermentation coupled with glycolysis only generates 2 ATPs. It is a very good table because if you look at this column, which is the terminal electron acceptor, this will explain the definition what I write on the blackboard. What is aerobic respiration? Final electron acceptor is oxygen. What is anaerobic respiration? Final electron acceptor is inorganic chemical other than oxygen. And the fermentation is also organic molecule. Most of the time is lactic acid. And we also call lactic acid is derivatives of pyruvate. And that's a very good table. Last one, pentose phosphate. This one I will be very briefly mention. Because you already learned so many cycles, we're not going to talk about everything. It's not about chemistry class like I mentioned. So we just go over some of these conclusions. Pentose phosphates will generate ribose, 5-carbon sugar, or deoxyribose, which is used for biosynthesized DNA and RNA. That's what we're going to talk in a few minutes. They will generate a reducing power is not NADH or FAD, it's NADPH. And they will generate precursor metabolites, most likely they use for anabolism process for generate DNA and RNA. So you can study by yourself for this cycle. It's not difficult understanding. Usually we in this class you just know some of these conclusions, which is good because we don't go over these too much. More important, you should know NADPH is a reducing power and they generate precursor material for DNA and RNA. Okay, that's why we will spending another chunk of time today moving to our next section, which is talk about genetics. There is a lot we could talk about that. Lots of exciting material we can mention. So we're going to move to the next section. Again, genetics section, if you're already taking genetics classes, that will help, but some of the explanation I will be explaining a different way, might be more detailed than that. If you already know, it's okay. If you don't know, it's a good time to learn again. If you already very well, it's like a review section for you, okay? So let me remove all these, then we move on to the new section. So we move on to genetics, microbiology, genetics. What are we going to talk? This is our outline. Central dogma. What is a central dogma? DNA going through replication by cell. I'm going through a second steps, generate RNA. This is called transcription. And finally, this RNA will generate protein. This process, we call it a translation. Now, not done yet. Lots of your textbook will say this could go back, called reverse transcription. We don't want to talk about that too much, okay? So here, since this is a central dogma, that will be an outline for this section. We will talk detail about the replication, transcription, and translation. And a little bit about transcription, translation, regulation, we using lacto-operon as an example. This is a very tricky stuff, like a maze, like a back and forth. We'll talk about that. So this is central dogma. This is what you have is a new version. But the older version, people don't talk about this. Back in 1950s, when people first talk about central dogma, they all talk about is nuclear gases go to proton. That is what the older version. Okay, in your textbook is a modern version, talk about central dogma. So let's talk about these one by one. First of all is the DNA replication, but before that, we need to go over some very typical experiments in the history. Why we want to talk about that? It's very interesting to know. Some of them relate to the microorganism, which means relate to this class. That's why we want to re-emphasize it when we talk about that. You will find those people really is talented and very amazing. Those are very cool studies. So we want to talk about the DNA first. Okay. When the people start to talk about the DNA, this is back in 1920s. People talk about some transformation principles. So what happened is, by the time they know there is some communication between genetic information between different bacteria, even different viruses, then they want to find what is that major thing to guiding those transformations. So that is called transformation principles. The idea coming out a very important and a very cool study is 1928 by F. Griffiths. Okay, what is that? This is related to our knowledge we already learned. Streptococcus pneumoniae. This is also called pneumococcus. Okay, some clinical name called pneumococcus. So if you see some people say this is the same thing, Streptococcus pneumoniae, we already learned that. Number one, community-acquired pneumonia in the United States. By the time, it is also called the damage. What's the major damage caused this from? Spanish flu. In 1912 started. Kills millions of people. And by the time, everybody wanted to find a vaccine. So they started to do the research. F. Griffiths is one of that. What he did? Streptococcus pneumoniae has one type, what we learned in the class, has a heavy capsule. Therefore, it becomes smooth. This type of Streptococcus pneumoniae is virulent, which means if animal or human being, especially animal, let's say a mouse, inoculated or infected with smooth Streptococcus pneumoniae, it will be died. But luckily, we have another type of the Streptococcus, which is, we call it a rough one. The rough one is non-virulent, which means the mice or the human being could survive, and we also call it a surrogate. So Griffiths did this very cool study. What he did? Okay, let's say I need to use this green color. Let me see where I find it. Okay. Smooth Streptococcus pneumoniae go to inoculate a mice. What happened? This mice is dead. For dead mice, we can draw another one. We can just draw like this. The mice is dead. Okay, let's just say a mice. So he had a second group, which used a rough Streptococcus pneumoniae. Then also go into a mice. What happened? This mice is alive, survived. So the next group, what he did? He used the same smooth Streptococcus pneumoniae, but heat injured, autoclaved, or maybe heated, and maybe boiled, whatever he likes to do. He go to inoculate the infected mice. What happened? The mice is also survived. Now we understand it because it's already heat injured. Then it's a very magic stuff comes. He using the same smooth heat injured Streptococcus pneumoniae. Okay, then coupled with a rough one. And then inoculated the mice. What happened? This mice is dead. This is dead mice. What this tells us? Something absolutely happened here. There is some genetic information causing the dead mice to transfer from the heat-injured smooth cell to the rough cell. So something happened. This is called transformation. So there is some genetic material there. And I tell you one thing, same as our Koch postulate. He even isolated in other place. He even isolated get Streptococcus pneumoniae. He's isolated. So that means this experiment is successful based on the Koch postulate. But what's the next step? We try to find what is that genetic material. That will be hard, to be honest. So he did a lot of study. Millions of mice, I won't say millions, thousands of mice have been killed. Because what his research is typically based on the animals, on the mice. Sorry, there's a lot of mice doing every time and then killed and then blended, centrifuge to get isolation. So he didn't make much progress during the 1930s. Then what happened? Unfortunately, in 1941, he was dead because he was bombed by the German bombing during the Second World War in his lab. And the rumor says he made some progress later on in the 1940s, but unfortunately he didn't get to that. He died. Okay. Then 1940s comes, 1941, 42. Everybody is busy, scared because Second World War is coming. However, there is a very smart people, a group of people. In 1943, in the United States, Avery, McCarty, and McLean, they do a group of study. What they did, instead of using tremendous amount of the mice, they did this in cubes. This is a progress. Because otherwise you're using so many animals and they're doing in virtual environments, which means they use the tubes to do. And what happened is they get isolation of purified DNA from this Streptococcus pneumoniae. Now what they did, you could look at these slides. Okay, basically it's a very simple way to do, not difficult. You have a tube. We're going to use rough sample. And then we're going to spread plated onto other plates. There is no bacteria there. Okay, what I'm going to do, the rough sample, I'm going to add a smooth sample extract. And then I'm going to go through the isolation. I got DNA. And I need to verify it is DNA. It is not RNA. It is not protein. So what we're going to do, in these treatments, we're going to add 3 enzymes, which is DNase, RNase, and protease. So what's the results showing? The DNase, nothing happens. The RNase, still DNA isolated, and the protease is also DNA isolated. So what this means? This means this genetic material, very possible, is DNA. And they say it's a purified DNA. Okay, so that's DNA. But I want to tell you, by the time when they figured out this molecule is DNA and this genetic information is DNA, it is not that exciting. It is not that fabulous. Why? First of all, people question how purified it is. You said you got DNA. It might be you have RNA. Maybe you have a limited amount of RNA. Maybe you have a limited amount of protein here and there. How purified it is? Second thing, which is if you look at the DNA structure, it is a very boring chemical. So let's talk about what is DNA. Why these people don't like it, say the DNA is a boring chemical. Okay, we want to talk about it. First of all, what's the full name of DNA? Can you tell me? That's a very easy question, but probably lots of people don't know. 2'